Welcome back, student, for the part 2 absorption and partition column chromatography. So we are learning the previous up to the various steps involved in column chromatography. That is, first is your selection of column, then selection of absorbent, preparation of absorbent. So these steps we are learn in the column chromatography part 1. In part 2, we will run the rest of the steps which are involved in column chromatography. The next step is preparing the column. So we are having two techniques for packing of the columns, preparing the column, that is packing of the columns. Either we are having the dry packing methods, dry filling methods, or wet packing methods, or also called as a wet filling methods. In dry packing method, the required quantity of absorbent is poured as a fine dry powder in column, and the solvent is allowed to flow through the column till the equilibrium is reached. In wet packing method, the slurry of adsorbent with the mobile phase is prepared and it is poured into the column. It is considered as an ideal technique for packing because the air attract in the column can be completely removed when you are using the wet packing columns. Before using column, it should be washed properly and dried to avoid interference of any impurities which is present in the column. The column should also be free from impurities and uniformly filled with the stationary phase before it is to be run. Steps in packing of a column. So generally we are placing a small glass wool or cotton wool in the bottom of column, a thick enough weight to prevent the passage of solid support, but not too thick that large amount of the air is trapped into the column plug. Fill the column 3/4 full of eluting solvent and drain a small amount of the column to remove any air bubbles from the glass wool. Tamp with the glass rod or some rubber tubing so that any air bubbles which is there can be removed. The glass wool must be free of air bubbles before you continue. You can use the powder funnel, add a thin layer of sand on top of the glass wool. Prepare a slurry of solid support, that is alumina or silica gel, by mixing it with some eluent and drain some of the eluent through the column. Then slowly add the slurry of the solid support till a particular height is achieved. Now the next step is the selection of mobile phase, that is called as eluent or solvent. The mobile phase or eluent is either a pure solvent or a mixture of different solvents. It is chosen so that the reaction factor's value of component of interest is roughly around 0.75 in order to minimize time and amount of eluent to run the chromatography. The eluent has also been chosen so that the different components can be separated effectively. You have to remember the basic principle of chromatography: like dissolves like. So whenever you are selecting the column eluents, you have to see that what is the nature of the compound, whether it is a polar or non-polar. The eluent is optimized in small scale um, spreads, often using the thin layer chromatography. We are using the thin layer chromatography as a basis for selection of the mobile phase. The solvent polarity is important. The polarity of solvent which is passing through the column affect the relative rate at which the compound moves through the column. If a solvent is too polar, movement becomes too rapid and later little or no separation of the component of a mixture will result. If a solvent is non-polar enough, no component will be elute from the column. Proper choice of an eluting solvent is thus crucial to the successful application of column chromatography as a separation technique. So the TLC will help us to determine the system for column chromatographic separation. The first step before you go for column chromatography, run the proper TLC and select the proper solvents. How the flow rate should be adjusted. A faster flow rate of the eluent minimizes the time required to run the column and thereby minimize diffusion, resulting in better separation. See the Van Dimter equation for this description. But in laboratory column chromatography, the flow rate is kept very minimum. A a simple laboratory column runs by gravity flow. The flow rate of such a column can be increased by extending. So in simple laboratory column runs by the gravity flow. The flow rate of a such column can be increased by extending the fresh eluent filled column above the top of the stationary phase, that is by giving some hydraulic pressure by increasing the amount of your mobile phase or eluent in the column, which can be decreased by tap control also. The better flow rate can be achieved by using pumps or by using the compressed gases. We can have a pneumatic pressurizer, that is using the air or nitrogen or argon gas pressure to push the solvents through the column. In patch chromatography, we are using this technique to increase the rate of flow of mobile phase through the column. The next step is introduction of the sample. The sample, which is usually a mixture of components, is dissolved in minimum quantity of the mobile phase. The entire sample is introduced in the column at once and get absorbed at the top portion of the column. From this zone, individual samples can be separated by process of elutions. The amount of sample to be added is critical. It should not be so high that it can saturate the column. The next step is the elution process. We are having three types of elution process. One is called as isochromatic elution, that is using single predetermined blend of the solvent mixtures having uniform composition until the separation is accomplished. The second is gradient elution, which uses continuously changing elution medium, helping in elution of more strongly absorbed component, simultaneously minimizing the tailing phenomena. So, in gradient elution, the composition of mobile phase is continuously changed during the process of elution, whereas in the isochromatic elution, the whole compositions remain constant throughout the process of elution. The third process is a fractional elution, that is also called as a stepwise elution, because only one specific solvent is used to separate one component of a mixture followed by another solvent for the other component. In that way, stepwise the component will get separated by using this fractional separation technique. Now, hints in packing of column. Sometime drain small amount of fluid from column, that will remove all the air which is trapped within the column. Tap the column with a piece of pressure tubings, that is rubber tubing, to help the solid support to settle properly. The column support must be packed gently until it is no longer settles. The final column of solid support should be 10 to 15 times as tall as it is as wide. So we are using 1 centimeter column tubing, then we should have the 10 to 15 centimeter height of the column. Never drain out solvent to the extent that surface of the solid support is exposed. This is the very important step in column chromatography that requires the frequent monitoring of column, that the level of column solvent should not fall below the level of your adsorbent. A small layer of sand is normally placed on top of column support to protect the surface, because whenever we are adding the new mobile phase or more eluting solvent, it may disturb your column packings, that requires some sand to be added on top of column, and it also should be below the surface of eluting solvent. After elution, analysis of the column eluents. So in column chromatography, this is one column which I have shown you in the figure. The components separated in column chromatography procedures are colored. The process of separation is simple. By monitoring visually, we can separate out the various components. But if the components are colorless, then we have to blindly separate the component using the various time fractions or volume fractions. More commonly, the component to be isolated from column chromatography are colorless. In this case, small fraction of eluents are collected sequentially in labeled tubes, and the composition of each fraction is analyzed by a thin layer chromatography. So before we go to the next part, that is factor affecting column chromatography, I will show you one video which will elaborate the process of column chromatography to you. So in column chromatography, the setup of the column chromatography which is used in laboratory is shown in this video. A small wad of cotton or glass wool is placed by a rod in the bottom of column. It should not be too large, otherwise more amount of air will be entrapped. Use the funnel to add small layer of the sand on surface of this glass vat. So this will support the stationary phase. Many times the sintered glass discs are available at this level. You can see it is 1 centimeter support in the column. Now we can add silica as a dry packing or as a wet packing. So generally we are using the wet packing method in which the silica is mixed with the solvent. Either you can use the eluting solvent or specific solvent. A slurry is prepared which is shaken thoroughly before it is to be added in column. Slowly it is added through the funnel so that a sufficient height of silica can be built into the column. So when you are adding, you have to see that every time you are shaking the beaker so that it will not get settled during the addition process. Add more solvent, shake it thoroughly again, and add the packing. When you are adding the slurry, you have to be careful that there should not be attrapment of any air bubble into the column or the slurry. You can tap the column with the pressure tubings. You can remove the excess mobile phase through the column, and you can reuse for adding the remaining part of your silica gel. Adjust the layer in such a way that it should not fall below the column heading. So now again, whatever remaining residues of silica gel, they are added in the top of column, and flow rate of your mobile phase is adjusted, and level should not fall below the level of your stationary phase. Open the cork to adjust the flow level. Once appropriate height of your stationary mobile phase is reached in the column, the ratio should be 1 as to 15 or 1 as to 20. I am using 2 centimeter column, so I am trying to keep 20 centimeter height into the column. In many cases, a sand is added before addition of solvent, a sample, but here we will add the sample before the sand is to be added. So the sample, it is to be dissolved into the particular solvent or the eluting solvent. Sufficient quantity of solvent is added. Care should be taken that no a high concentrated sample is introduced in column. The sample is introduced with dropper and it is distributed on top of the column equally so that even layer of the sample will be formed on the surface of the column. It can be achieved by rotating the dropper and adding drop by drop. After completing, again add a layer of the sand on surface of column. So this layer will prevent the further addition of mobile phase, or this will not produce disturbance of column. Add the mobile phase or eluting solvent so that a pressure will be built as a pneumatic pressure, and open the column tab so that the flow of material will start. Again, two steps will be involved, zone formation and zone migration. If the substance is colored, we can see the separation steps. We can see that the material is moving through the column and we can collect the fractions in the various tubes. So if the first fraction is not reaching to the end, you can collect the mobile phase through the first tube and reuse it for the further process of elution. Once the first fraction is collected, close the tab and replace the collection tube. We have to label the collection tube with the fraction numbers which we are collecting. We can collect these fractions in test tubes or we can collect it into the various vials. Now the solvent, the substance is reached to the bottom because we can see it as it is a color. But if it is a colorless, then you have to separate the fractions in 10 ml volume and every fraction is to be checked for its content either by TLC or by using UV spectroscopic analysis. So here all the fractions are collected in different tubes which is labeled for its fraction number. The process is continued till all fractions are removed from the column. The column should be colorless. If it is colored, then we'll stop this procedure. Every fractions we will analyze for its content by using the thin layer chromatography, whether it contains single component or more component. Thank you for this video which is available for the educational purpose. Now we'll see the next part, that is factor affecting efficiency of column. First is dimension of column. Efficiency depends upon a ratio of length to the width and sample. Column packing ratio, that is 1:20, is sufficient for effective separation. Also, the columns packing to the sample ratio is also important. So 1:100 is a sample to the column packing ratio. So if 1 gram is your sample, you should have 100 grams of your column packing. Next is your particle size. Efficiency increases with decreasing the particle size, but lowers the flow rate. So satisfactory particle size is 100 to 200 mesh, which will give you the efficient separation in column chromatography. The pore diameter, that is whites, pore diameter less than 20 angstrom gives effective polar adsorption. As we know, the temperature, which enhances the rate of elution, and the nature of solvent enhances efficiency of column flow rate. It is inversely proportional to the viscosity. So the temperature should be maintained at a fix so that there should not be change in viscosity. Then next is your packing of column. Uniform packing is essential, that we'll see in the Van Dimter equation, that packing size and shape is important, that will maintain equal voids and equal passage for passage of this mobile phase through the column. Now we'll see the advantages of column chromatography. It is a simple technique, can be used in both analytical and preparative applications. You know that analytical for identification purpose and preparative where you are actually interested in separated component. Any type of mixtures can be separated by column chromatography. If you are knowing the nature of substance and the mobile phase use, we can separate out the mixtures. Any quantity of mixtures can also be separated by changing the total length to diameter of the column. Wider choice of mobile phases is there in the column chromatography. We can use various numbers of mobile phases because in many cases the limitations are there in the use of the solvents. In preparative type, the samples can be separated and reused. Automation is possible in column chromatography. In laboratory also, the apparatus required is simple and cheaper as compared to your other instrumental techniques, but it has certain limitations that properly setting of columns require some technical skill and manual dexterity and takes some times. It is time consuming. More amount of solvents are required, which may be expensive as compared to your other techniques where very small quantity of solvents are required. Column chromatography is less foolproof than the paper chromatography and require constant attention while the experiment is being performed because the level of your mobile phase should not be fall on the surface and a fraction of 10 ml is to be collected every time. Automation makes the technique more complicated and costly, as we are seeing in this flash chromatography. The application separation of mixture of components can be done by using your column chromatography. It is used to remove the impurities or purification process. In many steps, we require that sample should be purified. Solid phase extraction process, that is by using this column chromatography we are doing. Isolation of active constituents can be done by using your column chromatography, either in small scale or into the preparative scale, that is in the large scale. You can see this column which is there in the figure. A very large quantity of the samples can be separated in such a preparative columns. Isolation of metabolites from biological fluids can also be done by using the column chromatography. Then estimation of drug in formulations or crude extracts of plant material can be separated by using column chromatography in the laboratories. Thank you and happy learning. See you in the next video. Thank you all of you.